Welcome back, folks. We're here with another one of the canine health episodes. In fact, this is a pretty important one because we're actually going to be talking about how to get the swabbing done correctly on your pup so that you can send it into Paw Prints and have them do the genetic testing for you. All right? So with that, I'm going to have Lisa start and explain to us how we need to go about this. All right, Lisa?
So Dan, did you wash your hands?
You know I did, too. You're kind of lucky,
good.
but I did.
Okay.
You want me to go do it again?
No, it's fine.
All
it's
right,
really
perfect.
important between dogs. We've handled other dogs today, and it's very important that between dogs you wash your hands so that we know that the DNA that ends up on one of these swabs is the right DNA.
So
from just
even
this
me
dog.
rubbing him like this, we can get what you call contamination, right?
Correct, because every cell of our
Wow.
body has DNA in it. So even the hair cells can contain DNA that could contaminate a swab.
Really? Well, that's important, guys. That's pretty important. Amazing.
So if I get, Cynean, if you could hold the bag, and all you simply do is break the seal and hand me the swab. What we want to do is swab the inside of the cheek, so the outer side of the cheek from the inside. And if you happen to drop the swab on the floor, you need to throw it away because the floor is dirty. It has other DNA. It has human DNA, maybe DNA from other dogs. So you need to throw it away and get another swab.
All right.
So we need, we give you 3 swabs, and we need a minimum of 2 in order to do our DNA testing.
But we should do all 3 and send them to you.
Yes, it's
All
best
right.
to do all 3 so we have plenty of DNA, so we
Okay.
won't run out of DNA. We can do our tests. Okay, so if you could please hold Kobe.
Okay.
What I'm going to do,
Pretty tight or just,
just so he doesn't move, because
okay.
this doesn't hurt the dog, but
Oh,
they're
hey,
curious.
sit.
And they don't really understand why we're doing this.
Sit.
So he's going to want to wiggle a little bit, and I need to roll the swab back and forth on the inside of the cheek for about 30 seconds. Now, when was the last time Kobe ate something?
Would have been last night.
Okay, good, because you want to make sure that your dog hasn't eaten for 2 hours. Okay, because again, we want the DNA on the swab to be that of the dog, not the dog food.
Oh, really?
Okay.
We're learning a lot today, guys.
So here we go. You ready, Kobe? Okay, this is not bad. You're going to like it. Okay, so about 30 seconds back and forth on the inside.
So does this swab maybe taste like chicken?
No, tastes like
Darn.
cotton.
With
And
your
then when
fingers,
it comes out,
you rub it back and
it should
forth.
look a little dirty, but not red. You want to make sure there should not be no blood.
No blood.
And then you're
So-
done with this one.
So it's all done.
It's all done. Put it back in its original container because it's a sterile container, and then you'll ship it back to the lab in this. So
All right.
Kobe's mouth is a little dry, which makes it, it feels sticky and it seems a little harder to get cells. So if you're swabbing a dog and their mouth seems very dry, you can let them get a drink of water and,
Okay.
and then swab again. So are you ready to try to swab, Dan?
I am.
Okay,
I
and
think it'd be kind of fun.
Cynean can hold Kobe's head,
All right.
and I'll hand you the swabs.
All right, so now, I'll just hold the lip up here, the upper lip, a little bit like this, and lay the swab against it,
Yes.
like that.
And then just roll it back and forth.
Roll it back. Oh, I see.
Yeah.
Okay.
So you're getting all the surface of the swab covered with cells. So usually what we found is the dog does really well on the first swab, and then the second swab they've kind of figured out that they can wiggle a little bit.
Yeah.
So you really need help. So it's really
Okay, so-
helpful.
So you don't leave that out in the air. As soon as you get done, you want to get back in the
Put it
container.
in the container. So
All right.
Cynean,
You
are
wanna
you
try
ready
it?
to try?
Yeah, I'm ready to
Okay,
try.
let me hold him then.
Okay.
This is not what I was expecting at all. I figured this would be
No, this
something
is
they
very
absolutely would not
This-
want to do, but it's pretty simple if you do it right.
Good
And then just
man.
twirl it a little bit.
Good man. Yeah, boy. That's my man.
So
you
now,
can see why it helps to have help holding the
Exactly.
dog.
you
It's a
couldn't do this by
two-person.
yourself.
It's
It would
a
just,
two-person.
guys, it'd be tough. You really need to have somebody help you hold your dog still.
Wow, pretty good.
Good boy.
Okay.
Now, if they get a little fussy, is it okay if you get saliva from the tongue, or is it you have to keep it on that cheek side?
We prefer that you gather cheek cells.
Okay.
We sometimes get variable results from people who send in their samples, and so the best way to get optimal results is to have someone hold your dog, to go on the inside of the cheek cell, and to twirl it and get the full surface of the swab covered.
So now, then we turn around and we put it back in the bag. I'm assuming it's a bag kind of like this?
Yes, the bag will look just like this, and
Okay.
you put it back in the kit that we provide you, and you mail it back to us, and the
Okay.
postage is paid.
So there's like, once it's collected and it's on the stick and in the bag, then it should come back to you as soon as possible, correct?
Yes, we prefer to receive it as soon as possible so we can start our testing.
All right, folks, again, we've got some absolutely fantastic information here, and I've got to thank Lisa for coming out to show us all of these very, very important parts. So if you have any questions, please don't hesitate to contact Lisa and her team at pawprintsgenetics.com.